Okay, this is a short video explaining the key points of nuclear import and nuclear export. So the whole process is summarised by this diagram, but to understand the process inside out you really need to understand this protein, RAN, in its GTP bound form and how it becomes in its GDP bound form. So we're going to cover that later. You need to understand that proteins are made in the cytosol. They have to bind the nuclear import receptor. That translocates them to the nucleus. And then proteins that are in the nucleus that want to get out bind the nuclear export receptor, and they then get out of the nucleus. This is the only way really of proteins getting into the nucleus and out of the nucleus. And this general process is not restricted to just single proteins. It's also involved in importing and exporting ribosomal subunits. It's also crucial for exporting of mRNAs and tRNAs. So if you block this process, the cell gets incredibly stressed very quickly. So this little cartoon just shows the endoplasmic reticulum, and an extension of the endoplasmic reticulum is the nuclear envelope. And within the nuclear envelope are these pores called nuclear pores. They're lined with a load of proteins which limit protein import and export out of the nucleus, and also limits mRNA export. So basically things can't get out of the nucleus through this pore unless they've got the right tag on them or somehow linking up with a nuclear export receptor. And this just shows the generalized structure of the nuclear pores. They're not just portals or holes in the membrane, they're holes in the endoplasmic reticulum extension, which is the nuclear membrane. And there's a whole series of different proteins that line these pores, and these limit what proteins can get in and what can get out. And the most complicated bit of the whole pathway is understanding why this protein RAN is crucial for nuclear import and export. And if you think about a protein in the cytosol binding to a nuclear import receptor, that's going to take the protein into the nucleus. Somehow you've got to prevent that nuclear import receptor taking the protein back out to the cytosol. So you've got to have some sort of directionality of the transport system so that the proteins just don't spend their life going backwards and forwards between the cytosol and nucleus and never actually do anything. They have to be displaced when they get to their final destination. Similarly, proteins in the nucleus that need to get out, they don't just want to be dragged back in again. You need some sort of directionality. And this is where this protein RAN comes in. And RAN exists in two different forms. In its GTP bound form, which is in the nucleus, and its GDP bound form, which is in the cytosol. And just think of GTP and GDP just like ATP. It's a unit of energy, and ATP bound is sort of the high energy form. ADP bound is the low energy form. Okay, so we'll not worry about how RAN is doing this as getting from the cytosol to the nucleus and back, but just for now, RAN GDP, its unphosphorylated form, enters the nucleus on the nuclear import receptor. When it gets there, we get transfer of GTP to replace the GDP, so that we end up with a pool of RAN GTP in the nucleus. And then at some point that will find its way in the cytosol, and whenever it gets to the cytosol it encounters an enzyme called RAN-GAP, and all RAN-GAP does is dephosphorylates RAN GTP into RAN GDP. Then this RAN GDP gets picked up again, brought to the nucleus. It encounters an enzyme called RAN-GEF, RAN-guanine exchange factor, and that effectively recharges it into its GTP bound state. So this cycle ensures that we've always got RAN GTP in the nucleus, and we've always got RAN GDP in the cytosol. And this is crucial when we get on to the next step of the directionality of the import and export process. And just be aware that this RAN protein is imported on the nuclear import receptor, it's exported on the nuclear export receptor, just like the other proteins, but as we'll see, it either travels on its own or with other proteins. So this rather complex slide explains the whole process that's going on. So if we look at point 1 of the slide here, we have got a protein here which wants to be imported into the nucleus. Let's say it's P53, because we know that goes to the nucleus. So P53 it's got a nuclear import signal, so that's lysine, 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 arginine, lysine. That allows this protein to link up with the nuclear import receptor and import in as a class of proteins. Then the protein is carried forwards. Ignore the fact that these arrows are double-ended. In reality, this is going in this direction. It gets the nucleus, and to prevent this protein, P53, being taken back to the cytosol, it encounters RAN-GTP. So RAN in its GTP bound form will bind importins, which are the nuclear import receptors. In doing so, it will displace our cargo protein, P53. It will sit on a different bit of the nuclear import receptor, change it, and the binding of RAN-GTP changes conformation of the nuclear import receptor, so it lets go of its P53, and then the RAN-GTP is carried back to the cytosol, where it encounters the enzyme RAN-GAP, and RAN-GAP dephosphorylates RAN-GTP into RAN-GDP. So the end result is our cargo started off in the cytosol, it ended up in the nucleus. When it got to the nucleus, it encountered RAN-GTP. Remember, RAN-GTP is abundant in the nucleus. That displaces the cargo, and then the RAN-GTP goes on the nuclear import receptor out, and that gets the RAN-GTP gets dephosphorylated to RAN-GDP, and that frees up the nuclear import receptor to bind more cargo. So it's directional. The P53 protein, in this example, can only get into the nucleus, it can't get dragged back out again. Now let's consider a different situation, a protein that's in the nucleus and it wants to get out. So this could be P53 once it's been used, and just in case you're interested, when P53 is monoubiquitinated, that exposes a nuclear export signal that is not normally visible. So P53 has done its work in the nucleus, it's become monoubiquitinated, that allows it to be exported. So what happens is this protein now recognizes a nuclear export receptor. So this nuclear export receptor has been imported into the nucleus empty. It then picks up RAN-GTP. Remember, RAN-GTP is abundant in the nucleus because RAN-GEF is there. Binding of RAN-GTP allows a protein with a nuclear export signal to bind to the exportin, which is the nuclear export receptor, and then the two proteins together on this nuclear export receptor are exported out of the nucleus, where RAN-GTP encounters RAN-GAP again. So RAN-GAP dephosphorylates the RAN-GTP into RAN-GDP. That changes the conformation so that RAN-GDP dissociates, and that forces the protein for export to be dissociated as well. So then we end up with a free nuclear export receptor that can come in and pick up a new cargo and a new RAN-GTP. So one question you might be asking is, how does RAN-GDP make its way into the nucleus to allow it to become phosphorylated, as we saw on the earlier slide? Because here we can see RAN-GTP going this way, and we can see RAN-GTP also going this way, but we've not really seen it coming in. And on the previous slide we see this cycle going on, which shows that RAN-GDP can enter the nucleus, and it obviously exits the nucleus on the nuclear export receptor, either with or without cargo. Now on this earlier slide it mentions that RAN-GDP is re-imported into the nucleus using its own nuclear import receptor. So that allows a constant supply of RAN-GDP into the nucleus, which then gets phosphorylated to RAN-GTP. So if we look at all the processes where nuclear import and export are involved, we need nuclear export of RNAs, both mRNAs and tRNAs. We need import of ribosomal proteins to build new ribosomes. Those are put together at the nucleolus. They link up with ribosomal RNAs at the nucleolus, and then the ribosomal subunits are exported using nuclear export out through nuclear pores. The sorts of proteins we're interested in, E2F transcription factors, any transcription factors really, they all are made in the cytosol and imported into the nucleus to do their work. Histone proteins are imported, as are your DNA polymerases. Then nuclear export of protein really comes about where you've got transcription factors which have been used, required, and they're no longer required in the nucleus, so they tend to get tagged somehow and then exported out of the nucleus to stop them from acting. And if any of you are interested, my research uses nuclear export inhibitors to effectively trap transcription factors and RNAs in the nucleus to stress cells. And when this happens, particularly in cancer cells, if you have a nuclear export inhibitor, will trap things like p53, all the transcription factors, and you'll block mRNA export from the nucleus, and this stresses cells really quite badly, prevents new protein synthesis, and induces apoptosis. So this is how I'm targeting this pathway as a novel anti-cancer mechanism.